Welcome back to our next chapter, that is biotechnology and its applications. आज part 1 देखते हैं, and in that we will find some tricks to go or introduce our subject of this topic. Okay, so let's have some tricks. We have seen the entire biotechnology chapter. Now where we can apply its benefits or application to mankind, that anthropocentric purpose of this biotechnology. So where we use the applications or where we apply the biotechnological benefits. So for that, just remember this alphabetical shortcut. Okay, the application starts here. GMO, that is genetically modified organisms. DEW, that is for the use of, D for diagnostic, E for energy production, and this W for waste treatment. Okay, diagnostic, energy production, and waste treatment. And then this FBT. Okay, F here for food processing. F for food processing. Here B for bioremediation. Bioremediation means cleaning up the waste place or pollution by using biological entity. That is called bioremediation. And T for therapeutic uses. So these are the major or broad applied areas where we can apply the biotechnological principles. Okay, now to do that, in biotechnology we do a lot of research. So the basic research for mankind welfare or production of different processes for product preparation go towards three basic research areas. Okay, so just like this trick, we have to understand and remember this, what are the basic three critical research areas in biotechnology. Number one, that is production of C, that is catalyst. Okay, we know the catalyst, the fastening process or higher production, what you call entity catalyst. That catalyst can be enzyme or can be microorganism or a cell. So first research area is diverted towards the production of such catalyst for industrial production process, where we can have a high quality yielding or a novel enzyme or a novel organism or modified organism for high production for mankind. Now first is catalyzed preparation in the form of enzyme or microorganism. Second is USP, that is upstream processing, that is optimization of condition. OC stands here, optimization of conditions. Now what is that optimization of conditions? The catalyst or that enzyme or that microorganism or cell we produced by research for production, what are its optimum condition, like at what pH it gives better yield, at what temperature it gives better yield, or what are the basic requirements, what are the ionic requirements, what are the different conditions, physiological conditions where which gives higher production. So that regulation or optimization is the upstream processing of industrial production, that is called optimization of conditions. Now the next, that is DSP, the third research area, that the product which we are producing has to be purified, and that we have seen in our last video. What is DSP? That is downstream processing, the final purification of the product or the industrial product to its purity level that can be then optimized, then different preservatives are added and went to the market for sale. Okay, so the third research area, that within very less investment, how we can purify the product of our industrial production. So three research areas. Okay, simultaneously production of, or optimize, a preparation of catalyst, either enzyme or microorganism, then optimal condition for that, and then finally purification of that product by downstream processing. Now let us see biotechnological applications in agricultural sector. Okay, इसके लिए एक simple trick याद रखना है with this alphabet ABA OA and GMC. Okay, now what does this ABA stands? That is agricultural or agrochemical based agriculture. Okay, agrochemicals based agriculture. Now what is this agrochemicals? These are actually this shortcut HFP. Herbicides, F for fungicides, P for pesticides, I for insecticide, N for nematicides. So use of these chemicals in agriculture for betterment of or production of high yield, that is called as agrochemical based agriculture. Now second is organic agriculture. Now what is organic agriculture? Basically organic or agriculture is centered in such a way that it will not harm these things, soil, environment and the people. What is the motto behind organic agriculture? That it should not destroy or spoil the soil, the environment there and the people there. So on what organic agriculture relies? It relies on ecological processes and biodiversity of that local area without any input of foreign things. That is the organic agriculture. That is the agriculture being done without harming the soil, environment and the people, which relies on ecological processes as well as the biodiversity of that area without doing any other input. Now the third one is the genetically modified crops. For the solution of all the problems in agricultural field is to go the modification in the crop at genetic level, that is called genetically modified crops. Now before going into these biotechnological applications in agriculture, let us understand कि वो green revolution भी तो हुआ था. Then if there was a green revolution, then that MS Swaminathan, Norman Borlaug, these people become so famous. Then why do we need these biotechnology in agriculture? Because that was the boon in case of research where green revolution was happened. But why these biotechnological applications are necessary even after green revolution? We have to understand some basic points. See, green revolution, although it tripled the production of vertical agricultural field yield, okay, the grains, everything production was increased, but it was not enough to meet the requirement of increased population. So population explosion was too high or on too peak. That's why this tripled food production was also not enough. Next, this green revolution was basically based on better management condition, that which crop grows in which season, what are the basic requirement in that season, and how to do manual work more smartly. That was actually the better management practices with the seed and agricultural practices. Next, there was lot of use of agrochemicals, this HF pin, that is herbicides, fungicide, pesticide, insecticide, nematicides. So that was actually not eco-friendly. Plus, if you go in poor countries, the availability of this HF pin, that is agrochemicals, is not possible. It's too costly for those people. So, in addition to that, whatever the conventional breeding methods are there, plant breeding methods, they are not enough to produce a high quality crop. That's why we have to move to biotechnological vertical ideas and research to apply in agricultural field to meet these what you call limitations, and for that we have to create genetically modified organism. Now what is genetically modified organism? For that we have a very simple trick. Okay, so how to remember this trick? First read the letters. F Bapu GM, for example, like Father Bapu, good man, like that. Okay, so here the F Bapu GM has a peculiar meaning in the definition of genetically modified organism, that is F for fungi, B for bacteria, E for animal, P for plant or any other organism, O, whose G for genes are altered by manipulation. Such organism is called genetically modified organism. It can be fungi, bacteria, animal cell or plant whose genes are altered by manipulation. Such organism is called genetically modified organism. Now what benefits we get if we have genetically modified crops in case of agriculture by using biotechnological practices? First of all, it gives the crop tolerance. Tolerance means the resistance, the ability to withstand against extreme conditions. Okay, so first benefit is tolerance under extreme condition. For example, cold, C for cold, D here for drought, S for salinity and H for heat. So tolerance in different agricultural conditions, different what you call physiological physical conditions or the conditions of the nature which are not suitable for that crop to grow, such a tolerance can be gained by biotechnological applications. For example, you can grow the plant where the soil is too much saline, too much salty, where there is no any chances of plant growth. But in that condition also, we can grow the plant by applying the biotechnological application. Even we can bring the plant from hot places. Okay, we can bring the plants from hot places and can grow them in our places. It is possible only by biotechnological application. When we do alter their gene, we manipulate them at a genetic level to make such genetically modified crops to grow anywhere to withstand this tolerance or this extreme conditions resistance, that is C for cold, D for drop, S for saline conditions, salinity, and H for heat. Second, rest in peace, that is REAP, that is reduced use of, R for reduced use of, I for insecticides and pesticides, because these are not eco-friendly. Hence, by reduced use of insecticides and pesticides is possible by using biotechnological principles. So second most important benefit is reduced use of insecticides and pesticides. So we have RIP in that keyword. Then resilience in mineral absorption. See, what we see, sometimes plant they grow and they utilize the nutrients, minerals so fastly, and after continuous cultivation on that agricultural soil, the soil becomes ultimately infertile because the depletion of nutrients or minerals there. By genetical application, हम plants को उस level तक modify कर सकते हैं that they start to utilize the minerals more efficiently, more slowly, and more gradually, but more beneficially. In that way, the depletion of minerals inside the soil condition will not be harmed or will not be vanished. So the next application is resilience in mineral absorption. Next most important is enhancement in nutrients value. The next important biotechnological application in agriculture is enhancement in nutritional value. For example, the golden rice or the vitamin A rich rice. Okay, so these are the benefits of genetically modified crops in case of agriculture, that is tolerance CDSH, then rest in peace REAP, that is reduced insecticide and pesticide, then resilience in mineral absorption, as well as enhancement in nutritional value. For example, vitamin A rich rice. Okay, that is golden rice. Okay, so in this way we can memorize the benefits of genetically modified crops. Now come to the highlight of today's topic, that is the BT cotton. Okay, now BT cotton, उसका long form होता है. It is based upon the name of a bacterium, that is Bacillus thuringiensis. Okay, ये एक बैक्टीरिया है और इसका जीन कॉटन के जेनेटिक सिस्टम में इंसर्ट किया गया है, इसलिए इस कॉटन को बीटी कॉटन कहा जाता है. ओके, सो हियर इट इज अ कॉटन वैरायटी whose genetic manipulation is altered by inserting a foreign gene from Bacillus thuringiensis. अब वो कौन सा जीन है? That gene is actually the gene that produces the crystal protein or just a prototoxin which is called as the cry protein. So that is a cry gene. कौन सा जीन है? That is the cry gene. So cry gene किसके पास से लिया है? That is being taken from the bacterium Bacillus thuringiensis. Okay, so Bacillus thuringiensis is the bacterium from which we have taken that gene, that is called cry gene, that produces a prototoxin. अब prototoxin क्या होता है? The form of toxin which when produced is in inactive form. The form of protein which is toxin, when produced it is in inactive form and it requires certain physiological condition to get activated to show its physiological effect. So prototoxin is simply an inactive type of toxin. So that cry protein genetically code for prototoxin, not directly toxin. Okay, that protein is also called as crystal protein. So this can be used to confuse in case of MCQs. Okay, so that cry protein is called as crystal protein or as well as it is called as prototoxin. Okay, तो ये बीटी कॉटन किसके अगेंस्ट, या बीट बैसिलस रेंजिनिस के जीन्स किसके अगेंस्ट इफेक्टिव है, अगेंस्ट दीज इंसेक्ट्स, दैट इज लेपिडोप्टेरans, दैट अगेंस्ट tobacco budworm and armyworm. We have to memorize this. Lepidopterans against tobacco budworm and armyworm. Coleopterans against beetles and dipterans against flies and mosquitoes. So lepidopterans, coleopterans and dipterans with their specific examples respectively: tobacco budworm, armyworm, beetles, flies and mosquitoes. Okay, so I repeat this: lepidopterans against tobacco budworm and armyworm, coleopterans against beetles and dipterans against flies and mosquitoes. इसको अच्छे से याद कर लेना. Okay, it can be asked in case of select the incorrect match type of questions or directly in case of select the correct option MCQ type question. Okay, now we understood that this is BT cotton, just not cotton, and it has a transgene. That's why it is a transgenic plant. Transgenic plant means such plant variety who has got a gene transfer from foreign species. So here the foreign species from which we have brought the gene and transferred into the genetic system of cotton seed, or what you call genes or DNA, that is called transgene, transfer of gene from Bacillus thuringiensis to cotton genetic system. Okay, so in this way this is the transgene, that is the transfer of foreign gene into the genetic makeup of the native plant. Okay, so this is active against this insect. अब हमको ये प्रोटोटॉक्सिन है ये तो पता है, that means it is produced as a prototoxin, so it is in inactive form. So how it is effective against this insect, that question definitely come at a logical level, that as we said this gene codes for inactive protein or inactive toxin or prototoxin, then how it is killing this kind of lepidopterans, coleopterans and dipterans. The logic is here in the mechanism. See, when insect attacks the cotton plant, what do? They feed on the plant. That means they eat the plant part, leaves, veins, etc. Now when they eat this, that plant part has to produce this cryprotein because it is in its genetic system. So these leaves and nails produces this prototoxin. The insect attacks the plant and feed on the plant. This prototoxin goes inside the gut of those insects in the intestine, stomach. Okay, now the pH in the gut of those insects is very high, very alkaline, that is 11 to 12. Now this is the condition where this prototoxin becomes actual toxin. So that inactive toxin becomes activated in this high alkaline condition in the gut. Okay, once that prototoxin becomes active, that is crytoxin, अब वो bind इसको करता है. There should be receptor to have a combination of reaction. So वो compatible receptors जो होते हैं, that is the cadherin receptors of the epithelial cells having brush border. We know there are different epithelial cells. One of them has the brush border. So उस brush border पे जो cadherin receptor है, this cryprotein goes and bite to that cadherin receptor on the brush border of the epithelial cells of the intestine. Now what happened? It directly dismantle the potassium ion regulation there. Okay, and the regulation of potassium ions is very much vital for the survival of cell, and because of this Bt toxin or crytoxin, there is a lack of potassium ion regulation that leads to lysis of certain epithelial cell, that leads to gap in the brush border mat inside the gut, and that causes lysis and death of that insect. So this is simple mechanism. Insect attacks the plant part they eat, that part consists of the inactive prototoxin. Then this prototoxin get inside the gut of the insect, get activated in high alkaline condition, that is pH 11 to 12, and then what happens? It becomes crytoxin. Where it affect by binding the cadherin receptors present where? On that epithelial cells having brush border. What do? They alter the potassium regulation, remember, and that causes lysis as well as gap in that brush border mat, and then there it leads to the death of that insect. Now which are those famous cry genes? Cry 1 in Roman, AC and cry to AB. For what you got this AB is active against the bollworm, whereas the cry 1AB against the corn borer. Okay, cry 1AC and cry 2AB against the bollworm, and cry 1AB against the corn borer. This also has to be memorized correctly. Cry 1AC and cry 2AB against this bollworm, and then cry 2AB, cry 1AB against the corn borer. Okay, so remember it correctly. It may confuse sometimes. Right, 2-3 times it will be kept better in the brain. Do remember it correctly. This part. Do remember correctly this part. Do remember correctly these matchings. It is very important bit. And then what are the benefits of this बेटी कॉटन? जब एक ट्रांसजीन या जेनेटिकली मॉडिफाइड कॉटन बनाया गया, what were the benefits? First of all, yield was increased. Obviously, if the insect is eating the plant and still there is no any chances of increasing the yield. But when that insects are being killed, they will not feed on the plant. Definitely plant will grow. So yield will increase ultimately. Second, the cost of production decreased. CPD means cost of production decreased. That is, whatever the agrochemicals we needed to kill those insects, bollworms and etc. There is no use of such insecticides and pesticides. That's why the cost of production was increased. Again, rest in peace, that is reduced insecticides and pesticide. Of course, it is EF, that is eco-friendly. So these are the advantages of this BT cotton. Yield बढ़ गई, cost of production decrease हो गया, no insecticide and pesticide use, hence it is eco-friendly. In this way, we have entered into the topic applications of biotechnology in agricultural sector with these tricks to memorize the different facts inside this topic. Okay, first, where are the benefits? Next, the critical research areas. Then in agriculture, what are the 3 different sectors we exploit biotechnological application? Then BT cotton, of course, its benefit. So in this way, just read these tricks, make their long form mug up, and then understand the topic correctly by writing once, twice, and solving the questions. Okay, so I wish you understand this, and I wish, thank you, thanks a lot.